Role of other excretory organs. In the list, we'll go through role of other excretory organ. So here it is, other than kidney, some other organs also, other than kidney, sorry, we have completed the kidney, other than kidney, lungs, liver and skin. Three are given here, lungs, liver, skin. So first of all, let us see the lung story. Then when we eat carbohydrate, we digest the carbohydrate in the form of glucose. In the intestine, whatever the glucose digested and absorbed into the blood capillaries of microvilli. From the blood capillaries of microvilli, through the blood, it reaches to all over the body. That means into the cells. So when this glucose reaches the cells with the help of oxygen, we break glucose and we get some energy, 38 ATPs. We call the process as respiration. In the mean period, we are going to form a excretory waste material. You can call it as metabolic waste. Metabolic waste of what? Metabolic waste of carbohydrate. The metabolic waste of carbohydrate is going to be carbon dioxide. So, the excretory organ which is going to eliminate this carbon dioxide is lung. This is the story of lung. So, what is the story behind the lung here? In carbohydrate metabolism, when the metabolic waste of carbohydrate is going to be carbon dioxide, an excretory waste material which is released in the carbohydrate metabolism is eliminated by lung. So in that context, lung is an excretory organ. Lung is an excretory organ. So for your understanding, we go with proteins also. We are already completed the issue of protein. When you digest the protein, proteins release amino acid. Whatever the amino acid formed here, we use for reconstruction of some various proteins in the body, that you can say DNA transcription, translation, a procedure occurs. In that, the waste material released is amine converted into ammonia. Based on the water availability, some organisms they convert it into urea, some organisms convert into uric acid. So in the protein metabolism, excretory waste is going to be released is either ammonia or urea or uric acid, whatever. Ammonia, urea or uric acid. In this series, you people need to understand that protein metabolism, waste material released in the protein metabolism is eliminated by kidney. Carbohydrate metabolism, whatever the waste materials eliminated by lungs. And the liver. What is the role of liver? Conversion of the ammonia into urea done by liver itself. We call the process as ornithine cycle. We say the procedure as ornithine cycle. So, this is the story of carbohydrate metabolism. We said the metabolic waste released in the carbohydrate metabolism is eliminated by lungs. Then application question, the dimensional question it is. And the excretory waste which protein metabolism. In protein metabolism, we form ammonia. That ammonia is converted into urea by liver and followed by urea is reached to the kidney, and kidneys are responsible for the elimination of the urea. So that is the total story behind this particular area. And let's now read in the text here. Our lungs remove large amount of carbon dioxide. How much? Approximately 200 ml per minute. And also significant quantities of water every day. So that is the story of lung, which we are completed in detail now. Liver is the largest gland of our body. Then liver, the another process is given here. Liver secrete bile containing substances like bilirubin, biliverdin. What is the story of them? We know after that 120 days RBC are going to die. So who is the graveyard of RBC? Spleen. So in the spleen, RBC hemoglobin will split into heme and globin. So heme is a toxic material. This toxic heme reaches to the liver. Then liver converts the heme toxic waste materials into bilirubin and biliverdin. You may call it as bilirubin and biliverdin also. The metabolic waste, bilirubin, biliverdin, is an excretory waste which is formed after the heme metabolism, Okay. So, these excretory waste released in the heme metabolism, the bilirubin and biliverdin. So, what is the role of liver here? Toxic heme converted into that bilirubin and biliverdin. What is the way? So, these particular excretory waste, liver through hepatopancreatic duct, it releases into the intestine. So, what is the way of bilirubin and biliverdin? They released out through the fecal matter. Okay, so that is the story of them. Along with that, we can say cholesterol and degraded steroid hormone, vitamins, and drugs are also released by liver. Okay, so this is the story of liver, largest gland, secrete bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormone, vitamins, and drugs. Most of the substances ultimately pass out along with the digestive vases. That's what we said here. Most of these are released out through the digestive vases. Through the fecal matter, they are going. Okay, now the smaller one, sweat gland. We need to know the component. Sebaceous glands. The component both are belongs to the skin. So, sweat produced by the sweat gland. Actually, what is the sweat function? When temperature increases outside, we are homeothermic animal. We need to maintain our body temperature constant. So, to maintain the body temperature constant, that particular sweat is going to be produced here. Sweat produced by the sweat gland is a watery fluid containing sodium chloride, small amounts of urea, and lactic acid. So, components of the sweat will be asked over for us. Sodium chloride, small amount of urea, lactic acid. So, primary function of sweat is to facilitate cooling effect on body surface. It also helps in the removal of some wastes mentioned above. That is the primary function is homeothermic nature. To maintain the homeothermic body temperature, sweat is much important for us. Along with that, they eliminate, sweat is going out. Along with the sweat, we are sending sodium chloride, small amount of urea, and lactic acid. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in the removal of wastes. So, these are the two functions. Now, sebaceous gland. What is the story of sebaceous gland? They eliminate certain substances like sterols, hydrocarbon, remember sterols, hydrocarbon, and waxes. So, through the sebum, sterol, hydrocarbons, and waxes. So, this secretion provides a protective oily covering for the skin. So, some amount of nitrogenous wastes are going through the saliva also. So, that is the total story of this particular role of other excretory organ. So, kidney, lung, liver, and skin. This is the total story. Okay. Moving on. Disorders of the excretory system. So, here it is number 1, uremia. In the disorder, uremia, which is highly harmful, may lead to the kidney failure also. Malfunctioning of the kidney lead to the accumulation of urea. Single line, urea in blood due to malfunctioning of the kidney, called uremia. First one. Okay, next, the second one. What is the next disease here? Here it is. Second one, renal caliculi. The next disease will be renal caliculi. Renal caliculi is the second one. Very, very important. Simply in our language, we can say kidney stones. Next, the glomerulonephritis. Inflammation in the glomerulus is said to be glomerulonephritis. Inflammation in the kidney is glomerulonephritis. So, need to talk about uremia here. When the kidney malfunctioning, if you see the malfunctioning of the kidney, that leads to the uremia. Urea is going to be accumulating in the blood. So, for that, malfunctioning of the kidney, the best solution is going to be the kidney transplantation. But till that time, this particular patient to be done with hemodialysis. What is the procedure in the hemodialysis? During the process of hemodialysis, the blood drained from a convenient artery, from one particular artery, will take it out. Let me take red color. Sequence question it might be. Convenient artery pumped into a dialyzing unit. What is the dialyzing unit? A fluid, dialyzing fluid. Okay, dialyzing unit. After adding anticoagulant like heparin, we add heparin. So, okay, so first remove, see this sequence. With the convenient artery, we'll bring out the blood. We add heparin, anticoagulant material. The unit contains a coiled cellophane surrounded by a fluid having the same composition of that plasma. Let me draw that coiled cellophane. This is the coiled cellophane. Okay. Then after adding, okay, next having the same composition of that plasma except the nitrogenous wastes, the porous cellophane membrane. The cellophane membrane is having porous, and this is called dialyzing fluid. This is dialyzing fluid. Okay. So, composition of dialyzing fluid is also going to be a bigger question for us. Composition of dialyzing fluid. What is the composition of dialyzing fluid? Dialyzing fluid composition equal to blood plasma. Particularly, we have to say equal to blood plasma except nitrogenous wastes. Blood plasma except nitrogenous wastes. Because we brought the parent because his kidney is malfunctioning and his urea is much accumulated. So, fresh dialyzing fluid does not contain urea. Blood of the patient contain urea, but fresh dialyzing fluid does not contain urea. So, this is what you people need to remember. And now, let's start. Some color I have to use, that's why I'm using. I can't use the blade color. Okay, let's do one thing. Let's use the blade. So, sequence lo first one, with the convenient artery, we brought the blood out and heparin is added to that. Then this particular patient's blood is going through the cellophane tube. From the cellophane tube, we have to bring it out. Okay, I'm bringing out. So then again what we have to do again, with a convenient vein, we have to bring in. Okay. So what is the difference? What we did in the mean period of time? So in mean period of time, the patient blood is containing urea. This patient blood urea containing, when it is passed through the cellophane tube, due to the differential concentration gradient, that to the dialyzing fluid, all this particular urea went over. That's why you don't find when blood is returning from the dialyzing unit without urea. Okay. What next? We'll bring it back again to the convenient veins. Okay. So that means here, you have to see here. Allow the passage of molecule based on the concentration gradient. As nitrogenous wastes are absent in the dialyzing fluid, these substances freely move out, thereby clearing the blood. Cleared blood is pumped back body through vein after adding anti-heparin. We added heparin in the initial time. So, anti-heparin to be added over. Okay, now let's try to understand the sequence. Bringing blood out from the convenient artery. Where it is? Here it is. Bringing blood from the convenient artery, number one. Number two, add and heparin, anticoagulant material. Number three, based on the concentration gradient, all that urea from the blood plasma of the patient dialyze in the urea flow into the dialyzing fluid. Okay. Next, that is number three, concentration gradient, difference between the dialyzing fluid and the blood of the patient, urea from the blood of the patient to the dialyzing fluid. Next, number four, blood is returned back. Number five, we add anti-heparin, and six is the vein. Okay, so this is the story of the story of hemodialysis. Let's do one thing. ఒక చిన్న వీడియో చూద్దాం. Then after we continue the story. Let's see the video. So this is the dialyzing unit. Dialyzing unit. So from the convenient artery, I am adding heparin. Blood is moving into the dialyzer unit. And then after the blood is brought back through the vein, we add anti-heparin. So the red one is artery, the blue one is vein. So continuously have to do means they do some grafting like so. So what is the mechanism here? The composition of the dialyzing fluid is equal to composition of the blood of the patient except urea. This is the physiology based question in that area. So from the patient's blood we are separating urea due to the concentration gradient difference. This one. This is the blood of the patient, more urea. Dialyzing fluid has no urea. That's why we are attracting urea. Majorly urea, but other waste substances are also there. So after removing this particular urea, the purified blood will be sent back to the patient through the vein by adding anti-heparin. There is no movement of useful substances here. Why? Because composition of useful substances, composition of any substance which we don't want to send is equal to the dialyzing fluid of the blood. So the patient blood and dialyzing fluid have similar concentration gradients. So not required for us, how much time it will take over, but definitely it's a painful process. So this is a story of hemodialysis, but finally this is a temporary technique only. This is a temporary technique itself because the kidney transplantation is a permanent solution for this. Ultimate method in the correction of acute renal failure is the kidney transplantation only. A functioning of kidney used in transplantation from a donor, preferably close relative ayi undali, to minimize its chances of rejection. Because otherwise their MHC doesn't match. MHC, a group of molecules which is very similar, relatives only have similar kind of molecule. We will discuss this in a immune system. MHC molecule matching they see. If MHC is different, rejection will be more. So modern clinical procedures have increased the success rate of such complicated technique, kidney transplantation. So finally they are saying that kidney transplantation is the only solution for this. So this is the role of other excretory organs and disorders of excretory system. Remember, this is a sequence story. Sequence question may come here. Okay. Then this is excretory products and their elimination.